My name's Olivia Vorius Lopez, and I did my Pecha Kucha on the immunologic tools mass cytometry, MHC peptide multimer staining, and activation-induced marker assay. The study looking at prolonged activation of resident T cells in nasal mucosa for COVID-19 used multiple different kinds of cutting-edge immunologic technology. The three discussed in this Pecha Kucha are mass cytometry, activation-induced marker assay, or AIM, and MHC peptide multimer staining. In this presentation, I will describe the ways in which each immunologic tool was vital to the study. Activation-induced marker assays were utilized in the COVID-19 T cell memory of the nasal mucosa study. This technology is used to identify and quantify T cells that had already been exposed to COVID before and proved local memory to COVID exists in the nose. AIM assays work by exposing T cells to specific viral peptides and then detecting surface markers that appear when the T cells become activated. This allows researchers to identify antigen-specific T cells without relying on cytokine production. Mass cytometry was also utilized in the same COVID study. This cutting-edge technology gave a very detailed snapshot of the immune response in the nasal mucosa. This uses heavy metal isotopes to separate cell types by their unique mass and even shed light on their activation states. It informed researchers that active T cells were present in the nose long after infection cleared. This was even up to 2 months post-clearance of the virus. This technology was able to help extract some of the most important data from the study. Next is MHC peptide multimer staining. MHC peptide multimer staining was also used in the study to target SARS-CoV-2 specific epitopes. This helped to identify the T cells that specifically recognize parts of the COVID virus. This testing also helped to identify the immune cells present in the nasal mucosa. MHC multimer staining is a technique used to detect antigen-specific T cells by using complexes of MHC molecules bound to a specific peptide and tagged with a fluorescent label. These multimers bind to T cell receptors, or TCRs, that recognize the presented peptide, allowing researchers to identify and analyze rare T cell populations by flow cytometry. The utilization of mass cytometry allowed the researchers from this study to analyze over 30 different proteins at the same time in a single tissue slice. This was in order to reconstruct a timeline of type 1 diabetes progression. Unlike traditional methods, mass cytometry provided both protein identity and the exact spatial location of each cell within pancreatic islets. IMC revealed that beta cells lose important protein markers early in disease. This is before they are destroyed. This would be very hard to detect with low parameter techniques, and this speaks to the importance and the efficacy of mass cytometry. It showed that cell function deteriorates gradually, not just via sudden cell death, something only possible to see when many markers are tracked simultaneously. The following are the references that I use to be able to make this presentation. Thank you for listening.